What's nice and hot in integrative neurology? You know, when I first became a vet, it used to be that surgery was one of the only options for an animal like this, or steroids, and quick responses without something invasive weren't readily available to practices like ours. What's incredible is this technology can take a dog from the former pictures to moving around and running and ambulatory, sometimes within 7.5 to 30 minutes in some cases. And that's just not explainable by old models such as the sodium-potassium pump or calcium ion channels. In fact, found that the more we tried to explain our quick results with the sodium-potassium pump, that the more it failed us in trying to explain the events that we were witnessing. In fact, in more research, we found that doubts about the sodium-potassium pump weren't even permissible, and that there seemed to be alternative paradigms to the sodium-potassium pump in physiology that we just weren't aware of, because there may have been some discrete fraud in the way that the data was presented to the sciences, especially the biological sciences. In fact, they said that lateral thinkers probably had no chance in our current system. It's interesting how the very current system produced a group of lateral thinkers that are changing the way science is done and presented by ordinary citizen scientists in combination with scientists from all over the world. And it all leads us back to water and how electromagnetic frequencies interact with water. Gerald Pollack found the same problems with water as he published The Ills of Science, showing how science had been taken over by interest in real science that could really change and cause revolution, just was not reported in the sciences like they should be. A search on YouTube of all places will reveal to you the genius of water, Gerald Pollack. Once you've seen the fourth phase of water and start to understand polywater and structured water, you'll never look at biology the same. A strong suggestion is to read all of his books, every single one of them, and fall in love with physiology all over again. A dive into Pollack will lead you to Gilbert Ling, who came up with the association induction hypothesis of physiology and seems to really be the unifying principle of physio as we understand it. His books can be found all over the world, published in multiple languages, and deal with water. Back to Gerald Pollack. Gerald Pollack has been instrumental in bringing forth knowledge of a fourth phase of water between water and ice. Here he describes it in his own words.
Here, and we spent quite a few years testing, and just to summarize, the surfaces that create these zones are many kinds of gels, many kinds of polymers. It was not definitive, but it gave us some idea of what might be happening. So we did the experiment, and what happened, what we saw, is that right after we put the suspension in, the particles were expelled progressively from this area. And the video stops here. If you look in the microscope, you'd see these particles dancing around in Brownian motion, but they would never enter back into this region. So this region looked like conceivably might be the extent of the ordered water region. And it was pretty large. We're talking 50 or 60 micrometers, which is substantial. So we call it the exclusion zone because it excludes particles, I'll show you later, solutes, or EZ, which is sort of easy to remember.
So to summarize, water, which is a dipole, arranges itself along hydrophilic surfaces and forms these zones of exclusion, large zones of exclusion, which excludes solute. That's solute over to the right. The water is organized in hexagonal sheets, one offset to the other, that forms large zones of exclusion that can be seen under electron microscopy. And this is orders of dipole layers of ordered structured water, which has different chemical properties and viscosity and negative charge. It's more viscous, the molecules are more aligned, it absorbs IR. It has a molecular structure similar to ice, almost as if it was one shifted to the right when it forms these long hexagonal sheets, and can arrange itself and stack itself similar to structure like DNA, which makes us question, is it the water that is really aligning and the nucleotides are aligning to it? These hexagonal sheets form this ordered structured water not just along living tissues, but also along inanimate polymer gels. Here we can see it along polyacrylate, and now Gerald Pollack will explain it as it works with Nafion.
You cut it to any desired shape that you like, and you plunk the sheet down in the chamber, as you see here, and you add water and microspheres and see what happens. Well, what happens is this, the same thing. This polymer does the same thing as the gel. And in this case, it actually extends out to approximately, typically 400 or 500 micrometers, half a millimeter. You don't even need a microscope to see it. You can see it with your naked eye. Very simple. Well, those are just two examples. By now, this basic finding has been confirmed by just part of a list, probably 20 or 30 different laboratories. And in fact, we were rather chagrined to find that it had been done 45 years ago and published in the Journal of Physiology by Green and Otori, who were looking at eye lens tissue and cornea, and they found the same thing, even the same size. So there's nothing new here. So there's nothing new here, except that this changes everything. Osmosis, as we understand it, simply cannot exist with a sodium-potassium pump, which really doesn't exist. What we're watching here is layers of the ordered structure water assembling and disassembling, and the solute coming in and only allowing osmosis when the ordered structured water layer decreases inside, and it's governed by electrical forces within the cell. If you read Gerald Pollack's book, Gel Cells and the Engines of Life, you'll find that it's not the old physiology anymore. Everything changes. It's all about the water and the wave and how phase transitions are going to govern all of biology. Our negative 70 to 90 millivolt potential is really about polymer gels and its interaction with water along hydrophilic surfaces. Enough juice to power up LEDs. You'll find that joints don't work the way we thought. It's not a friction model, but maybe an electromagnetic repulsion of two electromagnetic surface areas lined up against a really fancy polymer gel known as cartilage. Everything changes when you start taking in polymers and gels and water and how they form stable liquid crystals or unstable liquid crystals that are extremely electronegative along the hydrophilic surfaces. You'll find that when these electronegative surfaces collapse, then you'll have bone on bone, as it's the negative charges opposing each other that cause a net repulsion. And really, treating joint disease might be more about returning electronegative charges back to the surface of the joints. Gerald Pollack and his group found that water behaved very differently if you took these hydrophilic materials and rolled them into tubes like polymer gels and then ran this solute dirty water through it, that there was zones of exclusion that formed along the edges of these tubes and that the solute flowed through the middle of it. And basically this changed a lot of our ideas and everyone's ideas on how different solutes go through and function in the body. They also rolled up Nafion and found that it could transport water to a higher pressure, and that they had thought they had found the form of free energy because the Brownian motion kept moving the water through the tubes. A group also found that this is true in capillaries, that even at our smallest level, that our blood vessels never touch our vessel walls, as there's a zone of clear exclusion that doesn't allow the red blood cell to touch the side of the wall, and it's this ordered structured water that maintains this forward motion in the capillaries. Probably also true that radiant energy is another type of pump that pumps our fluid through our body. Someone in the lab left the light on and found that it was radiant energy, especially infrared and below, that charged water in particular ways. And it was the energy of vibration that imparts energy for building order and separating charges along hydrophilic membranes using radiant energy. The exclusion zones can be grown in orders of magnitude using radio and light energy and different forms of electromagnetic energy. In different books and publications on the physics of organisms, Dr. Mei Wan Ho introduced us to a whole new way of looking at biology and liquid crystals that are formed with the interface of colloids and water along hydrophilic surfaces, along microtubules inside the body. In Gerald Pollack's book, Water and the Cell, he described not just the death of the sodium-potassium pump, but the birth of
the association induction hypothesis and how crystals and gels work in the body to solve many of the paradoxes in medicine, such as how does edema form so quickly? What happens there? And it seems that water's constrained within the fascia until it breaks, and then these hydration shells begin to split apart once they're no longer constrained, and we have a change in phase from unordered structured phase to edema, pitting edema in the body within minutes. As the water disassembles, we found that we could use radio frequency to help change the phase of the disassembled water back into a liquid crystal, just like this. Here, 30 minutes later, the edema is gone from the previous pictures. How? Using radiant energy and the association induction hypothesis to create more ordered structured water and to coax the H2O into H3O2, a liquid crystal. Here we can show the edema going away after a 30-minute treatment. The X-ray on the left shows the edema before and the right after a 30-minute Sanawave treatment. We think this has huge implications on why our dogs were walking so early on in the videos quickly, and it has to do with mitochondrial and microtubule ordered structured water along living tissue. We believe that we can humbly learn from the smallest and simplest of organisms lessons about water and its flow through nerves that can teach us a lot about consciousness even in more complex organisms. We're finding that everything we were taught in neurology is the same. There are evidence of sonic booms in nerves and lipid membranes, of peroxynitrates breaking down into betanes, and that there's a whole new way of thinking about neurologic injury and edema, and about other alternative ways of bringing down edema and inflammation, restoring a liquid crystal layer around damaged nerves, and rapidly reinstill communication of what seems to be a liquid crystal breakdown, phase transition breakdown. The more we learn about phase transitions, and there's obviously a lot of evidence that there's phase transition activity in nerve fibers and cells, we're going to find newer and better creative ways, less invasive ways of treating neurologic disease in our animal friends. It seems like the more and more we look at phase transitions in biomembranes and in nerves, the more we'll find them. The more evidence we'll find of solitary shock waves along liquid crystals, and these liquid crystal layers acting as proton wires. The use of magnetic field stress to bring down inflammation has been around for about 50 years. Magnetic field stress produces heat shock protein 70, which has been found to be neuroprotective. We believe that the use of radio frequency in neurology is underrepresented, and we'd like to present cases of rapid neurologic rehab that takes animals from wheelchairs to running around like fools the way they were meant to, without the use of surgery, without the use of non-steroidal or steroidal anti-inflammatories in many cases, and provide alternative to our animal friends, which one day we hope will provide non-invasive cellular electroceutical options for human beings as well. The resonant pulsed electromagnetic field is ideal for the equine athlete. With increased blood flow, oxygenation, the secretion of heat shock protein 70 and other repair molecules, healing can happen deep from within and unlock excellence in the equine athlete. Nunez Therapeutics are non-invasive cellular electroceuticals for reduction of inflammation and return of normal function. The medical arts are changing rapidly. If you want to know the secrets of medicine, then think in terms of how energy, frequency, polarity, and vibration affect phase transitions of water in bodies on a nano and on a macro scale. Increased knowledge of phase transitions and their role in biology and how radio frequencies affect them are going to revolutionize medicine. What you saw here means nothing if it can't be reproduced in your animal hospital. Call us and ask us about our research licenses. We have an ongoing pain and neurologic study looking for animals with neurologic symptoms such as loss of function, pain, or anesthesia, or loss of proprioceptive deficits. We're here to serve. Nunez Therapeutics. Nice horse.